Hello guys, welcome back to Dr. Hughes Academy. Today we'll be discussing Pentose Phosphate Pathway, also known as HMP Shunt. That's your Hexose Monophosphate Shunt. Stay tuned. So the PPP, also known as the Pentose Phosphate Pathway, is also known as HMP Shunt. That's your Hexose Monophosphate Shunt. The glycolysis, which we discussed in previous video, I'll put the link in the description below, you check it out, is a catabolic reaction. So it breaks down glucose. However, HMP shunt is an anabolic reaction. So we have glucose being broken down to ATP through a process of what? Glycolysis. And this is a catabolic reaction. So the glucose can also be broken down through glucose phosphate to ribose 5-phosphates and other sugar phosphates. This HMP shunt forms NADPH. Remember, this is your HMP shunt. And it is what? Anabolic. So NADPH is formed in HMP shunt, while NADH is formed in glycolysis. So we have glucose in the blood, that's your blood vessel. We have a lot of glucose, and the glucose can move into the interstitial fluid. This is the interstitial fluid or tissue fluid over here. Now, this glucose has to enter the cell. Let's assume this is a liver cell. Let's say we have a liver cell over here, for instance. If this is a liver cell, what transporter do you expect it to be? This. GLUT2, right? That's correct. So this is GLUT2 for liver cell. If you don't still understand the glucose transporters, you can check the video on glycolysis. I illustrated it very well over there. So we have our glucose within the cytoplasm of the cell, and then the glucose is converted to glucose 6-phosphate by the enzyme glucokinase or hexokinase. Now, a rundown of glycolysis: the glucose 6-phosphate is converted to fructose 6-phosphate, and then fructose 6-phosphate to fructose 1,6-bisphosphate, which is now converted to glyceraldehyde 3-phosphate and dihydroacetone phosphate by the enzyme known as what? Aldolase. Then the glyceraldehyde 3-phosphate is converted to 1,3-bisphosphoglycerate, which is converted to 3-phosphoglycerate, then to 2-phosphoglycerate, and then phosphoenol pyruvate and pyruvate. And remember, reactions 1, 3, 10 are irreversible. The rest are reversible. So this glucose 6-phosphate can enter another parallel reaction. So the glucose 6-phosphate can be shunted into another reaction. The glucose 6-phosphate could decide to enter the glycolytic reaction, which is this reaction we described over here, or it could go into another reaction, which is the what? Pentose phosphate pathway. So the glucose 6-phosphate could be acted upon by an enzyme known as glucose 6-phosphate dehydrogenase, Whenever you hear dehydrogenase, what comes to your mind? NADPH, right? NADH or NADPH. In the case of the PPP, that's your pentose phosphate pathway, we are discussing NADPH. So NADP would be converted to NADPH by this enzyme glucose phosphate dehydrogenase. And what does it form? It forms 6-phosphogluconolactone. So 6-phosphogluconolactone is formed. Now, this NADPH is one major substance which we want to form. So this is one important product of the PPP, which is necessary for the survival of the cell. This is reaction 1. Then this 6-phosphogluconolactone would be hydrolyzed by the addition of water to form the sister molecule 6-phosphogluconate. And the enzyme involved is hydrolase, that's 6-phosphogluconolactone hydrolase or lactonase, which is 6-phosphogluconolactonase. Okay, let me put it over here. 6-phosphogluconolactonase. Oh, that's a long name. So the 6-phosphogluconolactonase would split 6-phosphogluconolactone to 6-phosphogluconate by the addition of water. And then this 6-phosphogluconate would be converted to another molecule known as ribulose 5-phosphates. This ribulose 5-phosphate is a 5-carbon molecule. What is the enzyme that converted 6-phosphogluconate to ribulose 5-phosphate? The enzyme is a dehydrogenase. So it is 6-phosphogluconate, let's say 6-phosphogluconate, permit me, dehydrogenase. So it's 6-phosphogluconate dehydrogenase. You had the dehydrogenase, what do you expect? NADP, right? So NADP plus would be converted to NADPH. Wow, another NADPH. So we're interested in this NADPH. Be aware that this NADPH is one of the major reasons we are undergoing this PPP. So we formed one NADPH and we formed another NADPH. And this was our reaction 2. This was our reaction 3. Reactions 1, 2, 3 are oxidative phases of the PPP. So they are oxidative phases of PPP, also known as the irreversible phases. If you can see, the reactions are irreversible 1, 2, and 3. Just a brief recap, the glucose phosphate was converted to 6-phosphogluconolactone by the enzyme glucose phosphate dehydrogenase using NADP to form what? NADPH. That's one NADPH formed. Then the second reaction was 6-phosphogluconolactone converted to 6-phosphogluconate by the enzyme hydrolase or lactonase. You can say 6-phosphogluconolactonase or 6-phosphogluconolactone hydrolase. So I prefer using hydrolase because there's the addition of water. You would remember it that way. So there's addition of water and then a 6-phosphogluconate is formed. This 6-phosphogluconate is converted to ribulose 5-phosphate by the enzyme 6-phosphogluconate dehydrogenase, and then another molecule of NADPH is formed. These 3 reactions are oxidative phases of PPP. So we've discussed the oxidative phase, that's your irreversible phase. Now we'll talk about the non-oxidative phase, that's your reversible phases of PPP. And I'll show you a mnemonic which you could use to remember it. The mnemonic goes like this. 3 grand exchange reactions for some PPP. So the 3 is for 3 carbon, and then it represents glyceraldehyde 3 phosphate. So the G over there represents glyceraldehyde 3 phosphates. Then the next one would be how many carbons? 4 carbons. So the next one would be 4 carbons, and that would represent E. That's your erythrose 4 phosphates. And the next one would be reactions. That's what? 5. Which would be your R and X. So R stands for ribose 5 phosphates, and then X stands for xylulose 5 phosphates. These two are 5 carbon. That's your ribose 5 phosphate and xylulose 5 phosphate. And then the next one is your F, which is what? Can you guess? Fructose 6 phosphates. And the last one is some. So this was 6. The last one is 7. So you can see we're going 3, 4, 5, 6, 7. That's your 3 grand exchange reactions for some PPP, for the reversible stages, right? So the next one is 7, which is pseudoheptulose 7 phosphates. So you can see we have 7, 6, 5, 4, and 3. So 3 grand exchange reactions for some PPP. 3 is glyceraldehyde 3 phosphates. The next one is 4, which is your exchange. That's erythrose 4 phosphates. Then the next one is 5. That's your ribose 5 phosphate and xylulose 5 phosphate. Then the next one is the F, which is fumarate. That's for the 6. And then pseudoheptulose 7 phosphate. Always remember that the number is represented over here. Coincidentally. That's good, right? So I'll show you how to use this mnemonic henceforth. So we have 3 grand exchange reactions for some PPP. Anywhere we see 3 is glyceraldehyde. Anywhere we see 4 is erythrose 4 phosphate. Anywhere we see 5, that's 5 carbons, is what? Ribose 5 phosphate and or xylulose 5 phosphate. Anywhere we see 6 is fructose 6 phosphate. 7, pseudoheptulose 7 phosphate. Cool, right? Yeah. So over here, we've already formed ribulose 5 phosphate. Remember, this is ribulose, not ribose. Ribose is what we discussed previously, but this is ribulose 5 phosphate. This ribulose 5 phosphate would be acted upon by an enzyme. So the ribulose 5 phosphate would be acted upon by an enzyme. This is a reversible reaction. So this ribulose 5 phosphate would be converted to ribose 5 phosphates. Now, ribose 5 phosphate is another molecule we are very much interested in. And that is why we are engaging in PPP. Remember, I told you that NADPH was one molecule we are interested in. Now, ribulose 5 phosphate is the second molecule we are interested in. So we'll put an asterisk on it. So we have ribulose 5 phosphate, and then the enzyme was isomerase. So to be isomerized from ribulose 5 phosphate to ribose 5 phosphate, and then another enzyme can come in and do something else, converting this ribulose 5 phosphate to xylulose 5 phosphates. The name of this enzyme is epimerase. So this is an epimerase. If you notice, we are discussing ribose 5-phosphate and xylulose 5-phosphate, which are the 5 carbon molecules we talked about there. So ribulose 5-phosphate was a 5 carbon molecule which underwent some form of isomerization. There was no change, or epimerization as this case. There is no change in the number of carbon atoms, right? So we have 5 carbon atom. Anywhere we see 5 carbon atom, you know it's either ribose 5-phosphate or what? Xylulose 5-phosphate. That's cool, right? Yeah. In the next reaction, we have a reversible reaction which involved the transfer of some carbon atoms from one molecule to another. Always remember with the mnemonic that we have something called alphabets, right? A to Z. Okay, so the one far back in the alphabetical order would always transfer to the one, closer to A. So we have xylulose and ribose. X comes after R. So Rx should transfer to R, right? Remember that the one far back in the alphabetical order does the transfer. So xylulose would transfer carbon atoms to ribose. That's another thing you should remember. Then the third thing you should remember the first one was 3 grand exchange reactions for some PPP. The second one is the alphabetical order, which we talked about. That's the one far back, transferring molecule or carbon atoms to the one closer to A. And then the third thing you should remember is that we have 3 enzymes, transcatolase, as your transcatolase, and transaldolase. So let's say TK and TA. Anytime we talk about transcatolase, we should think of 2 carbon transfer. Anytime we talk about transaldolase, we should think of 3 carbon transfer. So I'm pretty sure you've understood the 3 criteria for my mnemonic, right? The first one is 3 grand exchange reactions for some PPP. The second one is the alphabetical order. And then the third one is transcatolase, transferring 2, and transaldolase, transferring 3. So we have ribose 5-phosphate and xylulose 5-phosphate. Who should do the transfer? Xylulose, right? Yes. And then the first enzyme which comes here is your transcatolase. So transcatolase comes here, right? Well, let me just tell you, it uses the coenzyme thiamine pyrophosphate, for those that want to know, right? So transcatolase, it means that there has to be transfer of what? 2 carbon atoms. And then xylulose and ribose, which transfers? Xylulose. So xylulose transfers to ribose. If you remove 2 carbon atom from 5, what would you have? We have 3, right? So the xylulose would now form 3 carbon atom after transferring. That would be what? Glyceraldehyde. So that's your glyceraldehyde, 3 phosphates, okay? And then ribose obtained how many? 2 carbon atoms. That's your 7. 5 plus 2, that's 7. So it forms pseudoheptulose, 7 phosphates. Pretty easy, right? Cool. Now, between pseudoheptulose, okay, let's say pseudoheptulose and glyceraldehyde, 3 phosphates, these two would now undergo the next set of reaction. And then between pseudoheptulose and glyceraldehyde, which is higher or which is far back, is pseudoheptulose. S comes after G, right? So the pseudoheptulose transfers how many carbon? After your transcatolase, what comes next is transaldolase. So it transfers how many? 3. So if 7 minus 3 will give you what? 4. So we have what? Erythrose 4-phosphate. That's pretty easy, right? Erythrose 4-phosphate. And then after the transfer of how many carbons? 3 to glyceraldehyde 3-phosphate, it now becomes how many? That's your 3 plus, that's 3 here, plus 3 that was transferred, making it what? 6. So this gives us fructose. Remember, 6 is fructose, right? Fructose 6 phosphate. I'm sure you're happy. That's pretty easy. Fructose 6 phosphate. No need memorizing these pathways, right? So the last reaction is a little different because for the last reaction, we have some amount of ribulose, which was in the cytoplasm, that's ribulose 6 phosphate, which was then isomerized to xylulose 5 phosphate. And then this xylulose 5 phosphate then undergo the next reaction with erythrose 4 phosphate. So this xylulose 5 phosphate and erythrose 4 phosphate would now undergo the next set of reversible reactions. Remember, all these reactions are reversible. So it was transaldolase we had previously. Now we have what? Transcatolase. So transcatolase. How many carbon atom is transferred? 2. Who transfers to who? Xylulose transfers to erythrose. So xylulose is how many carbon atoms? 5. So xylulose 5 phosphate is 5 carbon atoms and it's transferring 2 to make it how many? 3. So the xylulose would now form what? Glyceraldehyde. 3 phosphates. And then the erythrose received how many carbon atoms? 2, because this is transcatolase, right? And it forms what? 4 plus 2 is 6. So it forms fructose 6 phosphate. Voila! We are done with the reaction. However, you should know that all these ones, like the glyceraldehyde, can go into where? Glycolysis. The fructose can go into glycolysis. This is another glyceraldehyde which can go into glycolysis. So depending on the body's need, these molecules, that's the sugar phosphates, could shunt into where? The glycolytic pathway. So this was our reaction 4, this reaction 5, reaction 6, 7, and 8. And these were the reversible steps or reversible phases of the PPP, right? Now let's discuss what happens to the NADPH and the ribose 5-phosphate. Already I've told you that there are some sugar phosphates that are shunted into glycolysis or glycolytic pathway depending on the body's needs, right? So one molecule is ribose 5-phosphate. And this ribose 5-phosphate can be used to form AMP. So the de novo synthesis of adenosine monophosphate requires what? Ribose 5-phosphate. Also, another energy carrier that ribose 5-phosphate is necessary for is NADH. Also, we have FADH. We also have coenzyme A. Even the NADPH we are talking about, the de novo synthesis, can be formed using ribose 5-phosphate. Okay? Now, this ribose 5-phosphate can also be used to form what? Nucleotides. Also, you can use it to form what? RNA. You can use it to form what? DNA. That's the synthesis of RNA and DNA require ribose 5-phosphates. Remember that if a cell is actively proliferating, it undergoes what? Replication, right? So the replication requires ribose 5-phosphate to form the backbone upon which nucleotides are fixed or attached. And you can also, transcribe the DNA to form RNA. And then the transcription of the DNA to RNA requires ribose 5-phosphate, which also acts as what? Backbone. And then the nucleotides require what? Ribose 5-phosphate. However, the NADPH has some unique functions. among which one of them is lipid biosynthesis, right? Like what? We can use it to form cholesterol. So let's just say cholesterol. We can use it to form fatty acids. We can also use it to form nucleotides. That's the NADPH. We can also use it to form nucleotides. So over here is the endothelial cell of the blood vessel. So this is the vascular endothelial cell. So let's bring out one vascular endothelial cell. Let's enlarge it. In this vascular endothelial cell, we have a molecule known as arginine. This arginine can be converted to what? Citrulline and nitric oxide in the presence of an enzyme known as nitric oxide synthase. And this requires NADPH. So NADPH is used to give off NADH. That's NADP rather. So in the presence of NADPH, arginine forms citrulline and nitric oxide using the enzyme nitric oxide synthase. And this nitric oxide is responsible for what? Vasodilation. So it helps the vessels to dilate. It also acts as neurotransmitters, and then it can also prevent platelet aggregation. So it prevents platelet aggregation. It is produced normally in the cells of the endothelium. So the vascular endothelial cells normally undergo this reaction to produce nitric oxide in order to maintain a patent vessel and to prevent platelets from aggregating unnecessarily. Okay. Also, we have phagocytes. So this is a phagocyte over here. And these phagocytes, or we can even say white blood cell, not only phagocytes, also white blood cells. So every cell that is involved in phagocytosis, right, they engulf the microorganism. So let's assume this engulfs a microorganism. And then the microorganism comes in. How does these phagocytes kill the microorganism? These phagocytes kill the microorganism by converting oxygen to superoxide. And this superoxide would now be converted to hydrogen peroxide, right? And the hydrogen peroxide can be converted to bleach, right? Your hypochlorite, that's your bleach, right? And this bleach is what kills the microorganism. But the point you should note is that the conversion of oxygen to superoxide requires what? NADPH. Okay? Now, let's go over to something else. So one other function of NADPH is that it is required for the reduction of glutathione. Glutathione destroys antioxidants normally. So let's assume we have a free radical, let's say hydrogen peroxide, in the presence of glutathione, that's a reduced glutathione GSSH, right? Is converted to water. So the glutathione reduces hydrogen peroxide to water and itself is oxidized to an inactive form as your GSSG. That's the inactive form of glutathione. Now, we need the NADPH to convert this oxidized glutathione back into a reduced form, which can act as the antioxidant. So, we want to convert it by, reducing this GSSG to GSSH. That's your reduced form or active form, so that it can now mop up more free radicals, right? And then we need NADPH for that. So for that, NADPH is used, and then it forms NADP plus, which can now go back to the PPP to be reconverted back to NADPH. That's cool, right? In this reaction, something can go wrong. There can be deficiency of this enzyme. So let's say this enzyme 1 can be deficient. That's your glucose phosphate dehydrogenase can be deficient. So when this individual has glucose phosphate dehydrogenase deficiency, what happens? It affects basically the erythrocytes, that's your RBC. Why? Because other cells have a way of, getting enzymes that can mop up free radicals. However, the red blood cells depend on glutathione for free radical mop up, right? So glutathione is the only antioxidant in the red blood cells. However, other cells have enzymes like catalase, superoxide dismutase, which can mop up free radicals and convert them back to water, right? However, the RBCs cannot and depends only on glutathione. And remember, I told you that glutathione requires NADPH for reduction to the active form. And when this enzyme is deficient, the glutathione cannot be reduced. And you remember, the PPP cannot continue. And we cannot generate enough NADPH for the individual. And what does this lead to? We could have a red blood cell over here. And remember, in the red blood cell, we have floating what? Hemoglobin. So this hemoglobin have sulfhydryl groups. That's the sulfhydryl group. So this sulfhydryl group are all these. And then in the absence of glutathione, that's the active glutathione, these sulfhydryl groups would be denatured, right? And then there would also be what? Lipid peroxidation. So the free radicals would destroy the sulfhydryl groups of the hemoglobin and also destroy the lipid membrane, leading to what? One of the things it would lead to is a change in the membrane, right? So the membrane becomes what? Rigid. That's one. And secondly, the hemoglobin will become denatured and then it will now clump and attach to the membrane. And this is called what? Heinz body. H-E-I-N-Z body. So this is called Heinz body. And since this red blood cell is rigid, that's the membrane of the red blood cell is rigid, as it passes through the small capillaries of the spleen, it cannot squeeze through. And then the macrophages within the spleen now engulf it and then hemolyze it. So the person comes down with what? Hemolytic anemia. So guys, we've come to the end of Pentose Phosphate Pathway. I'm pretty sure you enjoyed it. Thank you for sticking to the end. Do well to subscribe, like, put your comments below and share to your friends so they can benefit from this lecture. See you in the next lecture.